All right. Good afternoon, everyone. I'm Sandesh Marathe. I'm a postdoctoral scholar in the laboratory of Dr. Liza Makowski. I would like to thank the organizers for this opportunity to talk here at CTC 2023 on high-fat diet-induced obesity and identification of genetic modifiers of breast cancer using novel recombinant inbred strains. As a background of obesity, we can see here these maps from CDC, where obesity has a different effect on different populations. For instance, the Asian or white population are affected in between 20 to 35%, whereas if you look at the Hispanic or Black or American Indian or even Alaskan Native population, they are affected at a rate of 40 to 50% or even higher. Interestingly, the Mid-South, as Liza already mentioned, is affected at a higher rate in comparison to the rest of the states in the United States. And if you look at Tennessee, the Black population is predominantly affected by obesity, which is represented here by this diamond shape. Unfortunately, the burden of obesity falls mostly on women, where in the United States, around 77% of US women are overweight or obese. If you consider globally, again, obesity is rising, and once again, females are at a higher risk of obesity. There are 13 different cancer types that are linked to obesity, and hence it is important to understand how obesity affects the immune system and how it leads to cancer progression. Our lab mainly focuses on breast cancer, and we use the BXT model, as my colleagues have already shown, to study breast cancer. Our lab is experienced since 2010 to study breast cancer using the GEMS. But these mice are of a single genetic background, which is FEBN, and hence to make it more relatable to human population, we need to have a mixed genetic background, and hence we use the BXD mice. Based on this, the hypothesis is that system genetics can be used with recombinant inbred strains, which will help us identify genetic modifiers of cancer aggressiveness, and it will also help us study the response to exposures. We are excited to collaborate with our colleagues from UTHSC to study the BXD mice, which is highlighted by this review by Dr. Ashbrook and many others from the audience. We are excited that Dr. Makowski and others have been awarded both R01 and U1 to study the BXD mice. Important to my work, obesity is linked to cancer, and we are going to use system genetics to investigate the impact of obesity on breast cancer. For this particular study, to establish a link between obesity and cancer and how different genes impact the aggressiveness of breast cancer, we have selected different BXD strains. We have selected different BXD strains based on the work done by Dr. Suhita Roy, which I'll share on my next slide. Okay, thanks. Sorry about that. So we're looking at the data where the team has studied different BXD strains, which are shown here on the x-axis. And they have studied these mice, which were fed either a high-fat diet or chow diet. The high-fat diet is shown here in blue color, whereas the chow diet is shown in red color. We are looking at the body weights of these mice when they were subjected to these two diets for a period of 100 days. And we can see that the different strains have different susceptibility to weight gain or obesity when they are subjected to these diets. The gray bars represent the difference between difference in the body weights when these mice are either fed high-fat diet or if they are fed chow diet. So based on this data by Dr. Roy, we have selected different strains that are either resistant to diet-induced obesity or others which are susceptible to diet-induced obesity. The timing is important because the month of October is Breast Cancer Awareness Month. And as a reminder, breast cancer is the #1 diagnosed cancer in females, and it is also #2 cause of cancer death in women. And we want to use a mouse model, which is BXD. My colleagues have already described the BXD-BC model, where the C3 tag mouse is crossed with the BXD strains. And the progenies generate in females, we see various phenotypes such as we observe various phenotypes such as latency, multiplicity, volume, and weight. I won't be going into the details of these phenotypes, but I would like to focus on immune cells, mainly because obesity affects the immune system, and hence, since these different strains are going to show different susceptibility to high fat diet induced obesity, we are also interested in how the immune system changes in these different strains. So for this particular initial study, we have chosen 27 different strains, and we performed bulk RNA sequencing. And with the help of MD PhD student Jeremiah Holt and his mentor, Dr. Neil Hayes, we have used the bioinformatics tool ImmuneDecon to look at the immune cell composition based on the bulk RNA-seq data from these tumors. We can see that over here, we are looking at different strains of the BXDs. And based on the RNA-seq data, the immune cell composition and the immune cell composition of these different strains, we see that the strains have been placed differently, where certain strains show a hot tumor, which means that they are more infiltrated with the immune cells, whereas rest of the strains show a cold tumor, which means the infiltration by immune cells in these tumors is less. The hot tumors, or the mice that show hot tumors, lie close to the parent strain over here on the extreme left, which is the FVB mice. Another important thing to consider is the population of CD8 T cells in these tumors, which we have seen that in case of higher or larger size of the tumors, the CD8 T cell population is actually lower, which means that it shows worse outcomes in case of cold tumors. Using the T cell content of these tumors, we have used gene network analysis to find out the QTL, and we see that the QTL was identified at chromosome 11, where we see various protein coding genes. I'll focus on 4 of these just as an example, where we see that higher expression of GIP and MED1 shows smaller or lesser survival probability, which we have found out using the KM plotter in human patients. Whereas in case of other genes such as THRA or tensin 4, the survival probability is higher. So to summarize, we present a novel genetic model of breast cancer, which is a valuable tool to unveil genetic and environmental modifiers, such as obesity, that influence breast cancer. And as a take-home message, I would like to highlight these 3 points, which is we saw significant heritable variations in tumor traits in different BXD strains. Second, the tumor microenvironment immune cell differences are of human relevance. And lastly, there are varied responses to obesogenic high-fat diet, which points at the gene and environment interaction. So overall, genes and variants identified in this preclinical model and conserved across patient findings will ultimately help to improve patient outcomes. With this, I would like to thank the Makoski Lab. Very lucky to work with her. We would also like to thank our collaborators and the funding sources. I'll take any questions now. Thank you.